Hello everyone, I'm Shiting Li. I'm from the University of Michigan, Department of Computational Biology and Bioinformatics. Today I'm going to show a tool called METHYENRICH. METHYENRICH is a gene site and transcription factor enrichment tool. It's specifically designed for the DNA methylation high throughput sequencing data. As shown by the previous study, that the DNA methylation can either inhibit or promote the transcription factor binding to regulate the gene expression. On the left part of the slide is a study which used the in vitro method to evaluate the DNA methylation effect to the transcription factor binding, and they gave the percentage of the TF in the different scenarios as shown by the right part, that the DNA methylation can repress, promotes, or does not have any impact on the transcription factor binding. Although there are some old tools has been developed for differential methylation gene set enrichment testing, but almost all of them are designed for the Illumina B-chip assays. And we and others have developed some methods for the large size of genomic ranges gene set enrichment testing. However, the reduced bisulfite sequencing data and whole genome bisulfite sequencing data has specific properties that those methods are not performed well for those type of data. To begin with, we collect the data and to define a score to represent the TF binding sensitivity to the DNA methylation. And we collect both ataxic and ChIP-seq, and we use a motif to scan the peaks of the ataxic and perform the footprint. And also we scan the ChIP-seq and we obtain the potential transcription factor binding size and actual transcription factor binding size. And we combine those information with the same cell line whole genome bisulfite sequencing data, and ultimately we will calculate a table as shown in the below of the slides, that we will have a weight which represents the binding sensitivity to the DNA methylation for each TF. After we have the table and with the TF and their weights, and we are going to perform the enrichment testing in the gene level. So the first step is that we assign the user provided differential methylation to the TF-BS reference we have. So we consider both the motif region and also the flank region of this TF-BS. And after assign those DMR or DMC to the TF-BS regions, and we assign those combined regions to the gene. And we calculate a score for each gene represent the methylation effect, and we perform a generalized linear model for the gene set enrichment testing, and we use the number of the transcription factor binding size with CPG as adjusted parameters. Here are some preliminary results and some future work we need to do. So we draw the ROC curve and calculate the AUC to represent the performance of the METHYENRICH. And we collect the paired RSG and WGBS data from the spleen and the stomach. And we use the RNA enrichment testing results to represent as the golden standard. And here we can see the PolyEnrich is a method uses a large gene set of genomic ranges, and it uses all the DMRs. And the METHYENRICH only uses a small set of the DMR which has overlap of the transcription factor binding size, and it can perform as well as the PolyEnrich. And here is the reference include for 447 TFs. And we have some plan that we will build a more comprehensive TF-BS database, and we will cover around 1,200 TFs, and we want to characterize most of of the TF's methylation sensitivity. And we also need to implement a statistical method to extract each gene size TF perturbations from the methylation changes. That's all. Thank you.